Hi, welcome to a physionic long-form video, or welcome to a in-depth analysis of a particular study that we're going to be getting into, which is uh, going to be sort of the finishing touch to a series that I've been releasing that I have located in a playlist looking at a series of different studies related to the impact that saturated fat, specifically palmitate, has on our insulin sensitivity or insulin resistance. Both of those are the same thing on different sides of the coin of insulin and insulin signaling in our muscle cells. So this is going to be kind of the ending piece to that playlist. I think we've covered maybe 3 or 4 other studies on the topic, and we've kind of been building up to this study. And I'll certainly be having other series that are going to be related to this as well, specifically looking at other fats like stearate, for example, maybe looking at combinations of fats, combinations of saturated fats that you might find, because I know a lot of people ask those kinds of questions. So certainly don't think that this is the end-all be-all of content, as in we've covered a few studies now that we can dismiss it as we're done. We figured it out. We will not have figured it out. However, this study does give us a really, really cool clue into why some people may experience some of the detrimental effects that we've experienced, that we've seen with palmitate, with saturated fat, and why certain people may not experience detrimental effects. So we'll see if this study convinces you. I certainly have my own critiques of this study, which I'll be diving into as we go forward. But let me know your thoughts, and certainly also keep in mind that this is not the end. Just because it's the end of the series doesn't mean that we're not going to be picking up another series down the road and investigating even further. So my mind is still open on the topic, but we'll give you a succinct conclusion at the end if you want one. Okay, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and I hold my master's in exercise physiology, and I actually worked in a muscle metabolism laboratory for a number of years through my undergrad and then my master's program. And with those credentials out of the way, let's go ahead and jump into this study. So the question that we're going to be examining, if you'll allow me over here. So the question that we're going to be examining is, how does saturated fat, specifically palmitate, affect muscle insulin sensitivity in lean versus overweight individuals? So this is kind of giving us a clue into the distinction that might be occurring here. And the information comes from this particular study called Palmitate Acutely Induces Insulin Resistance in Isolated Muscle from Obese but Not Lean Humans. So this gives us, it technically gives us a conclusion by the title, but as I've said in many other pieces of content, just because the title says that doesn't necessarily mean the data corroborates what the researchers believe that it shows. So we're going to actually see that, and like I said, I do have a few critiques, which may be major and may change your mind if you actually agree with this title conclusion. All right, so to start off, what they did is they took, so some of the critiques certainly of the previous studies were that we've been looking at non-human cell lines, looking at L6 rat muscle cells. And while a lot of, or essentially all of that data was corroborated in human cells, this is going to be another study that's in human cells, but specifically not an immortal, what's known as an immortal cell line, but actual direct tissue from human beings. So what they did is they had a group of lean individuals or lean women. This is all in women, although I will say, although I don't know this for a fact, I will say that I do think that this would apply to men as well. But keep that in mind if that changes your perspective on it. But you've got these lean women or normal weight women, quote unquote, and overweight women. And what they did is the researchers is they took a sample, a muscle sample from each of these groups of women. So I think they had like 16 or something in each group, something along those lines. Let me quick check my notes real quick here. It seems that, yes, they had 19 lean and 16 overweight individuals that were part of this study. So a pretty large sample size, especially if you're going to be collecting cells, but they need a lot of cells, as one of my critiques is going to show. And what they did is they took these cells, and this is after fasting, so they're not doing any sort of special diet or anything like that at this point. And then they're plating these muscle cells, which are signified here, into, well, a plate. And then they give the cells the appropriate media, the glucose that they need, the fats that they need, the proteins that they need, all that stuff in this media to keep the cells alive. Then they took baseline measures of how these muscle cells react in general in a control condition, so just with regular media. And then they had another condition where the cells were exposed to a certain amount of palmitate. And then they had another condition, which I'm not going to be focused on too much, but I'll still report the data on here as well. They had another condition where they exposed the cells to palmitate plus adiponectin. I don't have a specific slide for this, but adiponectin is what's known as an adipokine, or a hormone essentially that's released from your fat cells. And this fat cell release hormone will bind to other tissues within your body and will actually make them more insulin sensitive. So keep that in mind, that adiponectin will combat insulin resistance. So those are, again, saying the same thing in two different ways. So adiponectin is typically considered a quote-unquote good thing, or good protein or hormone to have circulating in your system. So they expose these cells for, I think, like 4 hours or something along those lines to each one of these conditions, and then they looked at glucose uptake, which is essentially how sensitive are these cells to insulin, because they end up either adding or not adding insulin, and they end up testing the glucose uptake, so how much blood sugar or how much sugar is taken up from the media and how much is actually entering the cells. So this gives you a great measure of insulin sensitivity, so a direct measure of insulin sensitivity. And then another thing they wanted to look at was the insulin signaling, so the pathways, because if you've been following physionic for a while, you know that there's many different molecules within our cells that have to interact in certain ways for this to occur. So we're going to be looking at a little bit of data on insulin signaling as well. So that's kind of the general understanding. It's coming from humans. These are human cells, but the conditions that they're testing are in these isolated cells. All right, so let's look a little bit at the comparison between these two groups, because there's a few potential confounding variables. So I'm going to present these confounding variables or confounding factors that would essentially, what a confounding factor is, if you're not familiar, is a factor or a something that could get in the way of clean results of, we do this to the cells, we get this result, therefore this treatment that we added is therefore the reason why we got these results. There could be some confounding variable that would get in the way of that. So in terms of age, they're the exact same age. Usually there's a little bit of a difference, but there's the exact same age. Obviously, with body mass and BMI, they have to be different because one group is obese or overweight and one is considered lean. Although I guess you could maybe critique that this is on the higher end of lean or normal weight. I mean, I think there's definitely enough of a difference here that you can really tell the difference. Now, there's no statistically significant difference in fasting blood sugar levels. However, here's where we're starting to get into some of these potential confounding variables in that this shows that the fasting insulin levels were higher in the obese individuals, which would signify that they may be more insulin resistant, which turns out to be the case because they're HOMA-IR measures, which is an insulin resistance measure, was elevated in the obese individuals compared to the lean. So here, this is statistically significant on both counts. So this would imply, okay, well, maybe it's the insulin resistance itself that leads to the downstream effects. Now, I'm going to push back against that very same confounding factor in just a little bit, and you will have to then determine if you feel like that is a substantial enough reason to either dismiss the study or dismiss aspects of the study, or to say, okay, that's a good enough argument that it's not a confounding variable and the results that we see are appropriate. Free fatty acid levels are the same. And here we see another really interesting, or two different interesting things. So here's our adiponectin that I mentioned earlier that's released from the fat cells and increases insulin sensitivity. In the lean individuals, it's substantially higher than in the obese individuals. So this, this tells us some information right here, even that's independent of the study, that obese individuals or overweight individuals tend to have lower levels of adiponectin, which plays quite nicely with this insulin resistance, increased insulin resistance because of potentially reduced adiponectin levels. I don't think that it's necessarily one-to-one, like low adiponectin, therefore you have insulin resistance, but what it tells us is that there's a connection there, even if it's not necessarily causative, or that the entire insulin resistance is caused by the lowered adiponectin. It's just probably partly caused by it. And the other thing is leptin. So leptin being a hormone that has a variety of different controls, but one of the main controls is that it tells you about the kind of fat state of the cell or of the body in general. So if you have high leptin levels, typically it induces a point where your body is less hungry because your fat cells are constantly secreting leptin levels. However, what we find here is in the obese or overweight individuals, we have high leptin levels, and that's actually not surprising at all. Even though it's contrary to what I just told you, you would think that the overweight individuals, because they've been consuming so much, that they would have lower leptin levels, or that they would have these elevated leptin levels, and therefore that would stop them from consuming. Excuse me. But usually what happens, and this is from other data, that obese or overweight individuals, the idea is, the theory is that these individuals can have high leptin levels, but the receptors that leptin, will bind to are less receptive or are less sensitive, just like insulin. Just like you have insulin sensitivity, you can have leptin sensitivity. So the receptors will be less receptive to this leptin. So even with the continuous release of leptin in the bloodstream, it's not actually leading to changes in hunger or eating behavior or anything like that as a result. So even though these lean individuals have lower amounts of leptin, the leptin, once it binds to the receptor, actually has the intended consequence that it's supposed to have, unlike in this situation. That's a little bit of a side note, just to throw that in there. But the main one here is that this distinction here with insulin and probably with this adiponectin. Okay, so to open this up and to start getting into, this is the glucose uptake measure. So this is a schematic that I created to display what's going on here. So what are they doing? They're looking at these muscle cells, which is what they took from these participants, and they have a certain amount of glucose, and it's usually some sort of irradiated or some sort of tagged glucose or blood sugar that they can then track as it moves from the external sections of the cell as a proxy for like blood sugar levels, and how much actually enters the cells, okay? And here we've got our lean individuals, we've got our obese or overweight individuals, and the higher this value is, the more blood sugar is entering. So at basal, and here, just to explain this real quick, we've got a control, so no palmitate or no saturated fat added. We've got our P, which is the palmitate or saturated fat added. And then we've got our palmitate saturated fat plus adiponectin added. Now, as a quick added point, because there may be a question related to this, the palmitate levels that they added were physiological. So those would be the ones that you would find in your circulating bloodstream. However, the adiponectin levels were supra-physiological. So they were far above what you would find in a physiological situation. So this could be a reason to care or not care about how adiponectin affects these cells. And that's why I didn't really want to focus on it too much, because maybe it tells us a little bit of information, but can we really walk away from that with an entire change in our mentality? Probably not. All right, so now that I've explained that, so we've got our lean and obese, and then we've got our basal state. So this is without insulin added to the cells, so in both situations, and then with the addition of insulin, which is equal in both situations. So they add the same amount of insulin to the cells, and they find out what happens. So in the basal state, in the lean individuals, it doesn't matter what you treat them. Their glucose uptake, their basal glucose uptake is the exact same across the conditions, and it's the same for the overweight individuals. Now, what's happening, however, when they add insulin, and this is where they're actually stimulating insulin sensitivity, they want to actually measure insulin sensitivity by a measure of the amount of sugar that's taken up per molecule of insulin, because insulin is staying the same, so they're seeing how much sugar actually enters the cells. What they find is in the lean individuals, there is absolutely no change. So between the different conditions, so in the control condition, so usually you would compare these two versus the control condition, and they find that yes, there's a difference compared to basal, but there's no difference between these conditions, which would imply that palmitate does not have a negative effect on insulin sensitivity, specifically in leaner individuals. Or when I say leaner, I mean we're talking about a BMI of 24 here. So we're just talking like normal weight. I don't think these individuals are like bodybuilders or anything like that. They're just keeping themselves reasonably healthy. And this is a bit of a stretch, certainly. So I don't want you to think like these people are just like exercise fanatics and all this stuff. But it tells us just the normal weight individuals, which you would normally find. Now, on the other hand, again, in the obese or overweight individuals, the basal was the same, but once they added insulin, suddenly you started seeing these differences. So this mark right here gives us statistical significance versus the basal. So that's fine. That's exactly what we find here as well. However, in this situation, now we're comparing, that's a word, Nick, comparing the control in this white bar versus the palmitate, and what we find is that the palmitate is diminished in terms of the amount of blood sugar or sugar, glucose, that's taken up by the cells. Now, when they add adiponectin, there is a slight recovery of this glucose uptake. However, it's not complete. So we still see a diminishment in insulin sensitivity, which would imply, at least partially, that adiponectin is not the only reason why we have this reduction in insulin sensitivity. Now, here's where I wanted to push back against this confounding variable, if you allow me, with this HOMI-IR, because this would imply that these people are insulin resistant, and they likely very, very much are. However, in this scenario, looking at these cells, if you look at the y-axis here, we can see that we're looking at a standard amount. And if you compare the y-axis here versus here, they're the same, which implies that because they're adding the same amount of insulin, that there isn't this insulin insensitivity effect that's likely happening because of this y-axis being the same across both conditions. And it's not like they're adding more insulin to see an effect in this situation. They're adding the same amounts of insulin in both situations. So while these individuals may be or likely insulin resistant in some regard, at least it's not affecting these muscle cells that they've taken from these individuals. So it's not at the muscle cell scenario, maybe within the body. So, you could argue one way or another. There's a few different arguments you could make, but I think, for me at least, I think that this data shows that although the individuals as a whole are insulin insensitive or insulin resistant, at least a little bit, it still doesn't seem to affect these results. And I think that these results are most likely because of the palmitate addition and not because of their added insulin insensitivity that they have on top of things. So the effect is likely on them being overweight. Okay, so, well, I guess let me just to step back here. This right here, this data then implies that glucose transport or insulin sensitivity is unaffected by saturated fat palmitate and palmitate is the most common saturated fat known to man in the lean individuals, but it does seem to have an effect in the overweight or obese individuals, just to round that off. Now, I realize this looks, every time I show, insulin sensitivity graphs and stuff like that, I can just imagine people's eyes glazing over. But I'm hopefully going to do some justice in explaining this. We won't have to know all this information. We're just going to have to know a little bit. But here we are. So we're zooming into just a section of the cell. So here we've got our cell membrane. This line right here is our cell membrane. And here we've got our insulin receptor. We've got our insulin that's bound to our insulin receptor. We've got a bunch of different molecules. So all of these, most of these are proteins. Some of them are fat as well. So yes, you can have signaling through fat. And what they're doing is they're interacting with one another in such a way that ends up leading to this protein called a GLUT4, or a vesicle that holds GLUT4. This is the GLUT4 itself, which then moves from inside the cell. So here we're outside of the cell, here we're inside of the cell, and it will translocate, what's known as translocate, and move and integrate this GLUT4 into the cell membrane. This GLUT4 allows glucose or sugar into the cells. This is how we get this result right here. So the more of this GLUT4 enters or gets embedded into the cell membrane, the more of this glucose can enter from outside of the cell into the cell. Okay, so what normally happens, and I'll point out what's really important, but I'm going to go through the cascade, the insulin signaling cascade, is you have this insulin molecule that will bind to the insulin receptor, and there's a bunch of different things that happen. There's transactivation, all kinds of different things that ultimately happening, dimerization. Anybody who's interested in molecular biology will likely know about what happens to the receptor itself. But ultimately, the point is the receptor gets activated. This then leads to a phosphorylation event. Also, let's just, for the layman, just know an activation event. So the receptor is now active. It is now alert and can now be receptive to another molecule, and that other molecule is known as insulin receptor substrate. Then from there, insulin receptor substrate will be phosphorylated, which then leads to this cascading of events where you get this by PI3 kinase, which is another protein. And then this activates PIP2. It's kind of a funny sounding name. PIP2 will then be transformed to PIP3. You can see there's this cascade of molecules that have to change. And this then activates one of the key proteins that we're interested in, which is AKT. Now, the phosphorylation of AKT, which is activated in this scenario that we're not so much interested in TORC2. We're more interested in PDK. So PDK is then activated because of this whole cascade, which leads to the phosphorylation of AKT. Now, remember, again, phosphorylation means activation of AKT. In this scenario, not always, but in this scenario. So the phosphorylation of AKT then inhibits another protein known as AS160. What does AS160 do? Well, it's also known as like TBC or TDC, I think. But the point is that AS160 is a GTP ACE. What is that? It is a protein that will convert, in a very layman's terms, will convert this energy source. So we've always talked about energy sources as ATP. In this scenario, we're talking about energy sources as GTP, so guanine triphosphate. So this GTP will be converted by AS160 to a GDP. So Rab protein is attached to a GDP, and then it gets rephosphorylated to a Rab GTP. Now, in this form, as Rab GTP, it activates this GLUT4 vesicle to move to the cell membrane and deposit its GLUT4 into the cell membrane to allow glucose in. So normally what would happen is you have the activation of all this stuff. You have the activation of AKT, which inhibits AS160. It phosphorylates AS160 by, once it's been activated by phosphorylation, it inhibits by phosphorylation AS160. So it does the exact opposite. And then this inhibits this change from GTP to GDP. This means that this starts to accumulate, in that the RAB-GTP accumulates, which then activates more of this GLUT4 vesicle, which then moves to the cell membrane and allows more glucose in. So that would be normal insulin signaling. Okay, hopefully you got that. Please go through it again if you need to, and please give me tips on how I can explain that a little bit more, but keep in mind I can't spend half an hour explaining cell signaling before we get to all the other content. So hopefully that made some sense. But the big point is we're going to be focused on AKT activation, and we're going to be focused on the inactivation of AS160. Those are the two that we're focused on. Now, I still have it here so we can look at it as we go into this actual signaling. So what we're looking at is they're quantifying the protein levels. So these are proteins. So this is these smudges, as weird as they look, are actual protein levels. So we've got our lean individuals here. We've got our obese individuals here. And we've got a comparison between our different conditions. Again, our control, our palmitate, and our palmitate plus the adiponectin. And we're looking at AKT, and we're looking at not the phosphorylation of AKT. We're just looking at total AKT levels. Are the levels of AKT different in the cells between these two groups of individuals? And it turns out that no, there are no differences. So here in the lean individuals, we see no difference. They're the exact same. And in this scenario, it may look like to the eye, you would say, okay, well, if I'm comparing this to this, it looks lower. But statistically speaking, with these error bars and whatnot, there's no difference between these conditions. Okay, all right, so fair enough. So the total AKT levels are the same. So in that situation, that kind of goes against this difference in insulin signaling, or in this difference in insulin sensitivity with the palmitate condition. Now we're looking at the phosphorylation of AKT. So again, we're looking at phosphorylation as a sign of activation of AKT. And what we find is in the lean individuals, again, we see no difference between the conditions. However, when we look, and then I apologize, then again, we see that there's no difference. Now, I would be interested in actually looking at the raw data to see how close this got to significance, because it certainly looks that way. But again, the error bars are quite wide, or at least in this context. And I would also critique the fact that these blots aren't exactly great, meaning the blots are the measurement technique to get these protein levels. So yeah, these blots don't exactly look great, because if you were to compare this to this, it certainly looks a lot lighter than this. So this is what they have. I would say this is not great work, as in they should have had some added measures here to convince me that this was the case, that this is not different. But again, this is consistent with the previous data that we saw here, which is certainly more convincing than this data is. All right, but then something that they do is they end up adding another measure, which is another phosphorylation. So here we're looking at a threonine, what's known as a threonine phosphorylation. So an adding of a phosphate to the AKT at a threonine amino acid. And here we're looking at serine phosphorylation. And here we're seeing that, again, in the lean individuals, there's no difference. So this is consistent across all three measures. And here we're seeing that there is a statistically significant difference in that palmitate is vastly reduced by comparison to the control. Now, the addition of adiponectin sort of increased the level of AKT phosphorylation, but not much. So both of these are dampened by comparison to control. So this would imply that there's reduced insulin signaling through this serine AKT. Now, a critique that I have here is that typically you want what's known as a loading control. So you could argue, for anybody that's in molecular biology, you could argue that this is a loading control, so the total AKT levels, and that might pass. Clearly it passed these reviewers, and you could argue that that's okay. Typically what you want is a protein that doesn't change so that you can actually see the difference in the amount of total protein that they add. Because I'll give you an example. In this scenario, they could have added, well, I'll look at the change scenario. In this scenario, they could have added, let's say, 3 times the amount of total protein that they did in this scenario. And then that would be an unfair measure. That would be an unclear measure. In that, of course, this one's going to be lower. It might even be 3 times lower because they added 3 times less total protein. So when they measured just AKT of all the proteins, it was going to be 3 times less. So you want a protein that's independent of all these measures, that's consistent to show that there's no change. However, typically you don't see changes in total AKT. So in this regard, it's okay. However, this blot is, to be honest with you, trash. So I don't buy this data that well, but this data is certainly better. But again, I still have those critiques. Now, the critiques do increase substantially, and you could throw out the next piece of data when looking at AS160. And I'll touch on that in just a second after I've reported it. So this is the phosphorylation of AS160, which is, again, blocking AS160 activation, which means that the more you inhibit AS160, the more activation you have of GLUT4 going to the cell membrane. And again, in the lean individuals, we see that there is no increased, there or no difference, I should say, in the inhibition or phosphorylation of AS160. Again, these blots are not, visually they're not great. But statistically speaking, if they take all the data, because this is just from one experiment, if they take all the data together, they seem to show no difference. However, again, in the obese or overweight individuals, we do see that there is a dampening effect of AS160 with the palmitate condition, and even when they add the adiponectin. So adiponectin does not rescue this effect. Now, here's a problem in that before we had we could measure the AKT levels versus the 2 phosphorylation events. So we could look at the total AKT and just have a relative amount compared to the phosphorylated AKT. However, in this scenario, they do not have a total AS160 to compare against the phosphorylated AS160. In this scenario, you would absolutely want to have a loading control. You have to have a loading control. So in this scenario, I'm telling you, you can look at this data and say, I don't believe it, because they don't have a loading control. That is up to you as the researcher, as the reader, as the person analyzing this, to decide if you agree or disagree with what they're showing. And you would typically want to look at that in the totality of the data. Now, granted, I am leaning towards I believe them, but I desperately wish that they had done more for this. And this is, I think, a valid and powerful critique that they should have had a loading control. This is really, really important for this. But putting all of this together, in that we saw definitive decreases in blood sugar uptake or sugar uptake in the cells with the palmitate in overweight individuals, and we saw one measure of reduced activation or phosphorylation of AKT that was actually convincing, or not great, but it was there. And we see this effect. It leans just barely. It's like It's like the Leaning Tower of Pisa. It's barely, I don't actually, maybe the lean on the Leaning Tower of Pisa may be too extreme. It's it's like having a foot that's half an inch shorter than the other foot. You're barely leaning towards one side. That's where I am right now. So that's the data. Now, to give you a conclusion, a concise conclusion, if you have skipped to this portion and just skipped all the data and the explanation, let me say I have major qualms with certain parts of this study. So please keep that in mind as I'm about to report this very one-sentencey conclusion of the results of this paper. The saturated fat palmitate, specifically palmitate, reduces insulin sensitivity in overweight derived muscle cells, so from those individuals that were overweight, but does not have that effect in lean-derived muscle cells. So we saw that change in that difference between the two. And that was based off of some good data from the glucose uptake data, but the insulin signaling data was not great. We'll just put it that way. But if we just base it off of the glucose uptake data, the overall conclusion is still the case. But would I then jump from that and look at the insulin signaling data and say, oh, this is why. These are the molecules that are changing from palmitate exposure in overweight individuals that's leading to this effect? I think I would not feel comfortable with that, and I would dismiss that data for that. However, the glucose uptake, I think, is perfectly fine. Okay, folks, so there is kind of the end of this first series. And as I mentioned, I'm going to have multiple other series that are going to be looking at other saturated fats, are going to be looking at even more rigorous human studies. These studies, of course, tell us a lot more on insulin signaling specifically and give us more targeted effects on insulin sensitivity. But I've already started looking at research in humans. So I'll be presenting that as well in future series. But for now, this is where we will leave things. Let me allow this to kind of marinate in your mind that maybe certain subsets of the population may be unaffected by saturated fat exposure, at least in this scenario with palmitate. If you are lean, if you exercise, if you take care of yourself, but even if you don't, even if you're just generally healthy in your muscle cells, it seems like maybe your insulin sensitivity may not be affected. Now, a critique that you could have is, well, what if they had continued to take palmitate? If they were continuously exposed to palmitate, would that eventually beat down on the cell to lead to that insulin insensitivity? Maybe, but at least we can walk away from this saying that potentially there's a mechanism here. Not that we look at cell data, even though it does come from humans, not that we look at cell data and immediately say, oh, well, I'm convinced this is pure evidence, but alongside with all the other evidence that we've looked at, including human data, in actual humans, just doing measures in humans, which was a great paper to look at, it might create that distinction between leaner individuals or average weight individuals and people who are overweight. So for some people, saturated fat may have a slightly more detrimental effect than in other people. And if you decide to have a meal that's filled with saturated fat, or you decide to have like a week's worth of saturated fat, or just let loose, maybe your muscle cells can be robust enough and protect against the negative effects that you might normally experience. All right, so that is speculation still based off of preliminary data. I would say preliminary data, but we'll certainly continue to investigate this going forward. I keep adding caveats because I just know the comments just end up filled with cell data, can't do anything with this. It's like, no, context, apply context. All data I think has use as long as it's well done. but you have to apply context. That's what I'm trying to do here. All right, folks, with that, until the next series or until the next video, if you're interested in learning about other aspects, I'm going to be covering, what am I going to be covering? How carbohydrates affect our metabolism. That's some of the upcoming content, and I have some more stuff on mitochondria as well as some of the human data on saturated fats and unsaturated fats on insulin sensitivity. With that, I'll catch you in the next one. Thanks for stopping by, and till next time. Bye.